So my background is microbiology. So I studied for my PhD in New Zealand, where I studied microbes that can survive and live in hydrothermic hot pools. And since returning to Europe about 10 years ago, where I started as an astrobiologist, so applying my knowledge about extremophiles, which I didn't realize had any implications for astrobiology at the time, particularly when I was studying the microbiology of the inside of a cow, could actually apply to questions relating to astrobiology. So what I'm going to do in my presentation today is explain to you some of the work that I've been doing, but looking at how the importance of extremophiles are for astrobiology. To start this, what I'm going to do is give you an overview of microbial physiology, because you kind of need to know what cells are and how they work before we can get into the nitty-gritty of learning about these superhero microbes called extremophiles. So let's introduce why extremophiles are important for astrobiology. So, are we alone in the universe is one of the most fundamental questions of our time, and is the driving force behind the emerging field of astrobiology. And for thousands of years, we've had scientists and philosophers discussing about the presence of life, how it exists, and pondering and arguing about this for many times. And while there's no ubiquitous evidence of life that's yet been found beyond the Earth, we do know that there's the chemical building blocks for life have been detected in a wide range of extraterrestrial environments. So we have amino acids that have been detected on meteorites, and we've also found a number of them, which these are kind of the building blocks which we have for life as we know it. So if we want to look at this and understand this in more details, and with the number of missions that we have going to kind of potential habitable environments, we need to start to think about what habitability is and what are the limits of life, using Earth as an analog. Now, I'm looking at life beyond the Earth. I'm talking about carbon life form, because I think, we can start to actually fundamentally look at this in more detail. So to do this, we need to think about life in extreme environments on Earth and the limits of habitability as we know it. And most microorganisms that live in extreme environments are called microorganisms. And these are basically prokaryotic organisms, which you probably heard about previously from Laura, that fit into either the domains bacteria or prokaryote, bacteria or archaea. And we'll talk about those a little bit in more detail later. Now, microorganisms are ubiquitous in nature, so they can be found basically anywhere where we think there's liquid water. There are some exceptions to this rule. And so we're looking at places such as soil, hot springs, the bottom of the ocean floor, right through to the inside of the Earth's crust. And there's predicted to be about 40 million bacterial cells in only 1 gram of soil. And if we extrapolate that, there has been predicted to be something like 15 million bacterial cells on Earth. So they form most of the organic biomass that we have on Earth. So these microbes have an important role on Earth. They are known to be involved in biogeochemical cycling. So they can drive a lot of the organic carbon cycle and nitrogen cycle that we have on Earth. And they're also really important for looking at the origins of life, because we predict that some of the earliest life forms that we have was microbial about 3.5 billion years ago. And when we think about microbes, they're not all just one shape fits all. There are a variety of shapes and structures that we find. So we have these circular ones, which are called coccoids, which are quite common, and bacilli, which are kind of rod shapes. And then we have these kind of funky looking spiral looking organisms as well, which have been identified. And as you'd expect, called microorganisms, they're really, really small. They're at least below 20 microns in size. And here we have some images of some typical microbial cells under a SEM microscope. Okay, so it's really important to understand what a microbial cell is and how it actually functions before we start looking at how it survives in these extreme environments. So this is a typical bacterial cell. it's just a very simple one, but I think it's really important to get some of the key ideas to understanding what the cell function actually, what a cell looks like, and understanding how this has evolved over time. So if we look at these, if we look at this typical cell, this is here. So most of these, the cell is comprised of micromolecules, including nucleic acids, and we've got proteins and carbohydrates and lipids. So at the outside of the cell, some microorganisms have this kind of slime layer. It's called like the capsule, and it kind of consists of polysaccharides, and which can kind of somehow protect the cell, kind of protects the cell from the outer environment, and it also helps them to adhere to other cells and surfaces as well. They have these flagella, which can help with movement. And this again is really useful in the environment for swimming towards, like, for example, nutrient sources, or can help them move and to areas which are beneficial for their growth. So within the capsule, we have the cell wall. Now, the cell wall offers protection again and structure to the cell. So if we think about bacterial cell walls to start with, these are predominantly made up of peptinoglycan. And if you can see here on the diagram on the left, what we see is that it is made up of polysaccharide chains, which are cross-linked to form this kind of mesh, which provides a structural rigid for the cell and gives it kind of its characteristic shape. And again, this is really helpful in the environment because it can protect the insides of the cell from the outsides. We'll come back to this in a bit when we talk about the different domains of life, because this cell wall is really different, is one of the differentiations between the different domains of life. And when we think about bacterial, the cell walls, what we find is there's broadly two different kinds of cells of bacteria. We have gram-negative and gram-positive, which some of you may have heard about in the past. And this is a good way of kind of classifying microbes. And the way that we do this is we use a dye called, and we stain them with this violet color, and they're called gram-positive organisms. And these cells, what happens is the dye can keep the cells, so, and these gram-positive walls present this thick cell wall of peptinoglycan, which can actually keep adhering to the dye itself. And opposite to gram-positive, we have gram-negative, where they don't have this thick peptinoglycan cell wall. Instead, they have a single membrane. Instead of having like this thick cell wall, it's just a single membrane, single peptinoglycan layer. And then they have the outer membrane on the outside. And if we go back to the cell, what we can do then is we need to start looking at the plasma membrane. Now, the plasma membrane is kind of the key component which separates the inside of the cell to the outside environment. And although it's not like the cell wall or the capsule, it's not a strong makeup. What it is it's really important for cellular processes. It kind of acts as a semi-membrane, permeable membrane. So it allows the transfer of components and nutrients across the membrane. So it allows nutrients from the environment to be taken up by the cells, and it allows the secretion of waste products from within the cell to the outside environment. And it also is really important for producing energy. So what happens is that we can have something called a proton mounting force, so a gradient of protons across this membrane, kind of like a cell battery, which can drive the production of a molecule called ATP, which is like the cell currency of a cell and is used for cellular processes. So the plasma membrane is really, has traditionally, is kind of really important for these cellular processes and for the cell metabolism. So it's also commonly called the fluid mosaic model, which was discovered in the 1960s. And this explains how, this concept actually explains how molecules can move across the membrane. So the membrane traditionally kind of composes of two layers, like a biolayer of phospholipids. And here's an example on the left of what a phospholipid molecule looks like. It has a hydrophobic, aphilic end, which is the polar end, which actually is attracted to water. And then we have the hydrophobic tail, which does not like water, so kind of water-fearing. And this kind of phospholipid spontaneously assembles in such a way to keep the polar regions in contact with the aqueous environments, kind of forming like a sandwich layer. So you've got the polar lipids on the outside, which like the water environment, and then you've got the hydrophobic in the middle, a bit like a jam sandwich, I suppose. And although the structure itself, this phospholipid, isn't very strong, what it does have, these cells, particular example bacterial cells, have these structure called hopanoids in the middle of them, which kind of help to strengthen the cells as well. So you can see some examples of these in the cells, and you also have a number of proteins that can cross the membranes. And this actually allows for the transport of material from the environment to the cell and vice versa. Okay, so we know basically what is needed, what a cell looks like, but it's really, we now need to know about how this cell can grow and how it can reproduce. And for this, what we actually need, it needs to, inside the cytoplasm, one thing that we need to also mention is the fact that there's kind of this fluid, the cytoplasm is kind of this fluid jelly-like matrix. Inside that we have is where actually these cellular processes happen. So we have enzymes, activities, and where the building block happens. And what we do have within these cells are these things called ribosomes. And this is where we have home to where we have where the genetic codes form. So we take DNA molecules, as you can see here. Here's an example of DNA on the right here, which is the double helix structure that you're probably aware of. And it has these bases. So you've got your adenines, thymines, guanines, and cytosines, which kind of produces the genetic blueprint for the cells. And within these ribosomes, what we have is we have translation of this genetic code occurring, so that this kind of produces proteins and other structures. So this is kind of where cells need to make sure that they protect it, so that these processes can continue, whatever the environmental conditions. So we know what is needed for cell physiology, but what cells actually need. So when we think about cell life, this is for everything, for microbes to grow, they need three things. They need liquid water. Now, this is really important to ensure that cellular processes within the cytoplasm can exist. As I mentioned previously, the cytoplasm needs to be aqueous for these the movement of materials and processes to occur. It also needs to have the building blocks for life. So most macromolecules within the cell are made up of carbon, hydrogen, and oxygen. So we need to have those kind of as a source. And there are also other macromolecules, nutrients that are needed. So for example, we have phosphate, which is a key component in some amino acids, and also for nucleic acids. We have sulfates, again, which are important for amino acids as well. So an important thing we need to think about is metabolism. So when we think about, before we get on to think about extremophiles, some of these extreme environments that we know about, such as hydrothermic vents and hot pools, metabolism and how they grow is really important, something to consider. And for metabolism to happen, what we need to do, cells need to be able to convert energy to produce energy in the form of ATP. And now to do that, what we happen is a number of oxygen reduction reactions. So this is where we have the transfer of electrons from electron donors to acceptors. So if you look at the diagram here on the left, so we've got a nice reduced compound, we've got called compound A, and this is oxidized. So it loses as an electrons and it's coupled to B because they gain the electrons. And then what happens is that the compound A is oxidized and the compound B is reduced, and this releases electrons and protons into the environment, allowing for the formation of ATP. So all kinds of compounds can participate in redox reactions. So if we look at this redox tower that we have on the right here, we can kind of start to predict what reactions will happen in the different environments. So each reaction that we have has a different redox potential. So what happens if you look at this tower here, you have you have half of the coupled of the reactions in this reaction here, and from this we can kind of calculate some of these conditions. A good way to orientate ourselves when we look at this is to look for oxygen, because we know that this is really strongly oxidizing agent, and this is normally at the bottom of the tower. And we can start to kind of predict some of these environments. So, the most energy that would be produced is if you take an organic rich environment, such as glucose, what we use as humans, and we couple that with oxygen. This is a really highly efficient way of producing energy. So what happens is that they need to produce energy, and this is used then to produce ATP, which I've said before is kind of like the currency of the cell. And this is then used to produce simple organic molecules, which can be used as the building blocks for cells for microbial cells. Now, there's a number of different kinds of energy sources that a microbe can use, and this flow diagram kind of demonstrates some of those key points here. So primary production generally on Earth is driven by light. So most of it is from phototrophic organisms. So these are microbes that use photosynthesis. So we think about trees, we can think about algae, they all use energy from light. Now we can split that then down into organisms that need organic compounds and those that can fix carbon from carbon dioxide in the environment. So microbes that can fix carbon dioxide are called photoautotrophs. And for this, we have ones that can use oxygen. So these are oxygenic photosynthetic organisms. So we have plants, algae, cyanobacteria, and ones that do this anaerobically, such as photosynthetic green-blue bacteria, which actually do this without oxygen. Some organisms actually need organic compounds for this process. So for example, again, we have green non-sulfuring bacteria, which we'll come to in a minute, where they actually can just produce energy, but they need the organics from sunlight. But when we think of some of the most extreme environments on Earth, such as the hydrothermic vents and the deep subsurface, some of these environments actually light is not possible. Photosynthesis is not possible as the primary producers in these environments. There's no light source. So in these situations, what they do is organisms and ecosystems are based on chemotrophic metabolism. And this is where chemicals are used instead of light to produce energy. So again, chemotrophs, and this term can be split based on their ability to fix carbon. So for example, we have chemoautotrophs, and also in contrast to that, we have chemoheterotrophs. And a prime example of chemoheterotrophs where we use oxygen is humans and animals. So we use organic compounds and use oxygen as an electron acceptor to produce energy. And in contrast to this, we have microbes that don't use oxygen. So we have fermentative organisms, and we also have non-organisms that can do it using inorganic compounds as well. So let's look at these in a little bit more detail. So we have chemoorganotrophic metabolisms where we can use organic compounds as an electron donor and use inorganic electron acceptors. So for example, some of you may have heard of sulfur-reducing microbes, iron-reducing microbes. These are all organisms that can actually use inorganic compounds for anaerobic metabolism. So these are organisms that use it instead of oxygen. And in contrast to using the organic compounds as an electron donor, some organisms can use inorganic compounds So again, we're looking at things like ion oxidizers, so microbes that can utilize ion oxidation or sulfur oxidation, or even hydrogen as a form. So methanogenesis is an example of where you can use hydrogen to produce this. And when we think of it in the terms of beyond the Earth, so we're thinking about places like Mars or the icy moons, we're looking at some kind of, we think and we postulate that we could have chemolithotrophic or chemoorganotrophic metabolisms in these environments. Okay, so we know that they can use these different compounds, inorganic or chemical, to produce energy, but how do they actually do it in the cell walls, in the cells? And this is a really important process within microbial cells, is because they need to use, because the formation of energy actually doesn't happen in freely in solution. What actually needs to do is it needs to be coupled to atoms or molecules. And this generally happens within the plasma membrane. We have something called the respiratory chain. And here what happens is we have the transfer of electrons from one acceptor to the next. And energy is produced during the way. And we have this gradient producing of this protomotive force, where we have a gradient of protons occurring across the membrane. And these protons can drive the production of ATP through using the ATP synthase that we, as you can see in this diagram here. And this allows the production of ATP. So to now, I've just thought to discuss microorganisms as a collective, but as you've heard from Laura, that we've got these different domains of life, and microorganisms, particularly the extremophiles, that we're looking at extremophiles here, and most extremophiles fit between are either bacteria or archaea. There are some examples of eukarya. We have the alkaline shrimps, and we also have tardigrades, but predominantly we're looking at the domains of bacteria and archaea. Now, the main difference between archaea and bacteria, something that is key to extremophiles as well. What we know about bacteria is, we know they're unicellular. Archaea is a relatively new domain. It used to be grouped with bacteria, and there are obviously some similarities between bacteria and archaea. And some of the most of those would be that they're unicellular and the cells lack nucleus or general organelles. But the main differences between them, and I think this is something we need to think about when we think about extremophiles, is the cell membrane. And this is really important for microbes in extreme environments. So you can recall the general structure of cell membranes in bacteria, where they have this phospholipid bilayer. And in archaea, what they have is the composition is different. So rather than having, in the archaea, they have these isopropene links instead and they have these links. So they have in the archaea, they have these ether linkages, which are really strong compared to the ester linkages in bacteria. And they also have these side chains as well in the archaea, which can help with the rigidity of the cell membranes as well. Another difference between archaea and bacteria is the fact that archaea have these, certain archaea are known to have these kind of S layer. So like bacteria, archaea have the cell wall, which is semi-rigid and provides protection. And while cell walls normally contain this protein glycan, what we find with archaea is that some of them have this kind of S layer component, which again can protect the cells. So there's a number of ways that they could do it. They can either be anchored to the plasma membranes, for example, in diagram A, or they can just be connected to the pseudomurin layer. So we now have a kind of basic understanding of the physiology of a microbial of microorganisms. We know what they look like, what their structure is, how they grow, what they need as an energy source, and how that can differ between environments. So we've looked at it from a nutrient perspective. Now let's look at this in the perspective of what we know about life. So if we look at this diagram here, that kind of, it's a simple diagram which really nicely indicates what we know about life so far. So this is taken from a paper by Everest and Schupp back in 2007. And he's basically here, they've mapped on some parameters such as pH and temperature. It's a simple diagram looking at just two parameters, pH and temperature. What I like about this diagram is they put it in a kind of, it's easy to contextualize. So for example, you look at the pH scale, we look, it's got the pH battery acid at about just under pH 1, right through to household bleach. And again, we've got water boiling through to freezing. So if we look at the middle, at this green diagram in the middle, which shows familiar life, this is kind of where humans and animals can grow and live quite happily. So we know that the pH of blood and saliva is just circumneutral, and we know seawater, again, it's not too extreme. We can go swimming in the sea, everything's fine. But when we start looking at the microbial communities, we can start kind of pushing these boundaries that we know. Some of the most fundamental kind of influential work in extremophiles was carried out back in the 60s and 70s, studying Yellowstone National Park. And here we've kind of been able to push our knowledge of boundaries. So the orange square kind of shows the environmental conditions which we know have been identified in this environment. I like the fact that they've put coffee on this diagram. So if you're drinking a cup of coffee at this moment while listening to this talk, just think a microbe could live in that coffee. What they've also shown here, though, is also you can see that even though these parameters are more extreme than what we're used to, that is actually life that have been identified. So we have microbes like the sulfur bullous, which is very hot, that can survive under heat and acidic living environments. But since this initial work that's been done in Yellowstone, we can start pushing the boundaries in different directions as well. Acid mine draining, and for example, Rio Tinto has brought together a lot of information about the effect of pH and temperature on microbial diversity as well. So some of the most acid living environment microbes that we have been found about pH 0. And we can compare that to stomach acid. We know that microbes can live within the stomachs of humans. If we go to other extremes where we look at alkalis, we have places like soda lakes, such as Mono Lake, which are really alkaline in pH. And we've known that we can isolate microbes from these environments. So let's look at some of the environments where we know that microbes can be found. And I suppose over the past century, the boundaries and conditions where life has been found has expanded, and we've started to push these boundaries. And there's been a whole load of experiments and expeditions. And as our ability to sample these environments has increased, with our technologies, so has our knowledge of habitability and the limits on Earth. So life has been found in a whole array of different environments. And this kind of diagram here demonstrates some of these environments where life has been found. So we've got polar environments, we've got right through to deep sea sediments, through to we found microbes in nuclear containment facilities, microbes that can survive the highly nuclear environments that have been identified there as well. So there's been a whole range of environments where microbes have been isolated. So they're not all just natural environments. Again, there are also human-made environments where some extreme environments have been found and detected. So classifying extremophiles isn't brand taking, it's quite a simple procedure. So extremophiles is acid loving. So we have a number of terminologies that have been developed to look at this. So we have acidophiles. So these are microbes that live in really high acidic environments, so normally below pH 3. And at the opposite end of that, we have alkaphiles. We have the traditional ones, which I'll talk about in more detail. So microbes that can survive at extreme temperatures, pH, radiations. And then we have some more novel ones. So we talk about plesiophile microbes. So these are microbes that can grow at high pressures. And as our technologies have developed and we've been able to go to the bottom of the ocean understanding and isolation of these microbes has evolved over time as well. So let's look at some extremophiles. So let's look at these in detail. So if we look at take temperatures as an example. So if we go down to -20, right up to 120 degrees, we can map out where different groups of microbes can optimally grow within these environments. So you notice that it's kind of like a bell shape. So the top of that is kind of the optimum condition in which those microbes can grow. So if we start right at kind of the below zero, we have the cryophiles. So these are microbes which can grow, they don't just require, they need to have these conditions to grow. They can't grow above 0 degrees. We go through to the psychophiles, and then the mesophiles are kind of microbes that can grow at between 20 and 45 degrees. And then we get to the other spectrum where we have your thermophiles right through to hyperthermophiles. Again, it's very similar with what we have for pH. We have the acidophiles at one end of the spectrum and the alkophiles at the other as well. And from an evolutionary perspective, extremophiles are really diverse. So here's a general picture of the tree of life, and what we can find is they are found throughout. So if we look at the table here, we've got the hyperthermophiles, which are kind of clustered together, but also we look at psychophiles, and the blue, which you can see is a myriad of them across the domains, and also pHs. So you can see that so from an evolution perspective, extremophiles are really interesting to study. And because we have most of these hyperthermophiles seem to be kind of clustered together at the root of the tree of life, we're actually really important, extremophiles are really important for looking at the origins of life and also life on early Earth. There's a lot of work that have been looking at seeing, the impact of this. We can use these hyperthermophiles as a potential origin, looking at the potential origin of life. And the flexibility of the microbial genome has actually allowed them to do this. So because it's such a small genome, it's allowed them to evolve over these different extremes. Okay, so let's look at this in a little bit more detail. So temperature is a key parameter for that we will start with. Mostly what we know about extremophiles stems from microbes at extreme temperatures due to the work that was done at Yellowstone many moons ago. And what is interesting about temperature is that microbes have no control over it. So with some parameters such as pH and salinity, they're able to regulate their internal, to ensure that their internal cell, they can control and evolve, so they can control those conditions. But temperature is one of those parameters where they have no control. So the pH external to the cell is what it is internally to the cell. So if we take, for example, we look at a thermophile here, we've got Bacillus stereophilophilus on this graph. It grows optimally at 60 degrees. And this means that their enzymes that they have within the cell optimally are activated at 60 degrees. And temperatures below that, we have a decrease in enzyme activity, and it slows down metabolism, and anything above that can actually denature their cells. So if you think about, you take an egg and you cook an egg, and that is what happens when you denaturize proteins with temperature. It completely changes the way that it looks and reacts. And this is really important for when we consider temperature in relationship to physiology. So what we find is that microbes can grow over relatively defined temperature gradients. So they kind of have that optimum temperature. As I said, for Stereophymophilus, it's 60 degrees, and they only have a range of about 30 degrees in which they can able to grow, due to the fact that their enzymes can only be activated over a certain amount of times. Also, generally what we find, if you look at the diagram on the right of the membranes, temperature has a huge impact on the plasma membranes and membrane permeability. So at low temperatures, what we have, so about 0 degrees, phospholipids don't have much energy, so they are really closely packed together. And this makes the cells pretty rigid. And as we increase the temperature up to 45 degrees, the permeability increases and the phospholipids start to move around and they have more energy. So they're not, so no longer are they pack together really strongly. What they're actually doing now is they're making the membrane partially permeable, which can be problematic. So above 45 degrees, what you find is that the membranes become really problematic because they become they become really permeable to solutes. And this can impact cellular processes and also the rigidity of the cell. But although this is a problem in theory to these conditions, to this physiology of a cell, we do know that microbes can live in these extreme environments. As this diagram here demonstrates on the right, at the bottom, we have microbes, extremophiles, that can live at low temperatures as well as high temperatures. So the question is, how do they adapt? Well, let's look at hyperthermophiles to start with. So there's a number of mechanisms that we know. Hyperthermophiles we know exist. They've been found in a number of environments. They're predominantly been isolated from environments which are above. They normally are isolated environments that are above 80 degrees or so. And in a whole range of environments. And what we know about them is that hyperthermophiles can grow above 80, and the most highest record that has been shown is 122 degrees. And although that they need these high temperatures to grow, they can actually survive at lower temperatures. And this is really important. So we can find that they can be transferred from one place to another quite easily at colder temperatures. But then they can become activated when these high temperatures are available. Now, due to the solubility of oxygen at these high temperatures and the presence of reduced gases, most hyperthermophiles are actually anaerobic organisms. So they don't use oxygen, instead they use some kind of chemolithotrophic metabolism. Although there are obviously exceptions to this. So if you look at environments such as sulfonic fields where they're exposed to air, there is an amount of oxygen that actually allows aerobic metabolism to occur. So how do these organisms adapt? Well, there's a number of mechanisms that we know that that microbes have adapted, these hyperthermophiles have, that have allowed them to survive and grow in these extreme environments. So I think the first thing we need to think about is the DNA. So in theory, DNA would be deneutralized at these high temperatures, but what we find is that the DNA we think has a higher composition of GC, so guanine and cytosine, and we think that this actually has an impact to contribute to the sustainability of the DNA, which is really helpful and to support this and to ensure that the integrity of the DNA is there. A major point is that the enzyme efficiency of some of the proteins within the cell have been adapted. So the composition of these enzymes are adapted so that they are active at these higher temperatures. So these enzymes are stable and efficient under high temperatures. They also we also find that they have these kind of heat shock proteins, which are best known to one of the cells which are upregulated, so overexpressed in high temperatures, and these kind of protect cells from damage from temperature increases as well. And one thing we need to be aware of at these high temperatures is, they're really metabolically active because, enzymes work at higher temperatures. And what happens is that the membrane becomes really impermeable, to protons. Most hyperthermophiles above 100 degrees are actually archaea rather than bacterial. And this is because of the archaeal cell wall. So the archaea are actually are able to maintain proton permeability at these temperatures due to the composition of the cell wall. And this is called a process called homeoproton permeability adaptation. So this kind of helps, so the structure of the archaea actually lends itself to being hypothermic, surviving under these hypothermic environments. And if we look at the opposite end of the scale at the psychophiles, psychophiles again have been found extensively in the environment. These, when we think about the environments where psychophiles could actually exist, there's over about 70% of the Earth is covered by ocean, and by definition, microbes that live in these ocean environments are psychophiles. So most of the temperature at the lower levels are between 4 and 5 degrees. And we also have polar regions, again, which we can study, which are really low in temperature, where we're looking, and these constitute a further 15% of the Earth. So psychophiles, again, they make up a lot of environments where psychophiles could actually grow. So on the diagram here on the right, what we can see is a diagram that came from a paper from Panoply a few years ago, which has demonstrated that what the limits of these psychophiles can be. So we think by definition, microbial growth of a psychophile is between -20 degrees and 10 degrees, but there has been work done which has measured activity as low as just 39 degrees. So in the paper, what they've done is they took soil samples from different environments, from Siberia, taken these soil samples, and they've measured over a year CO2 and C14 labeled to see if the microbial communities within these environments at these temperatures could actually metabolize. And from these results, we were able to determine that they could measure activity as low as 39 degrees. So how do microbes within these environments, how does this happen? How can they survive? Because, at these cold environments, cellular freezing can occur. If you take water, the inside of the cell, it's a liquid fluid environment. If it was pure water, you could freeze it, you could see it would produce these kind of icy structures, which can result in cellular damage. So to compensate that, what psychophiles do is they lower the freezing point of the cytoplasm by accumulating cellular solutes such as glycine and sucrose within the cell, and this kind of lowers the freezing point of it, so it kind of protects the cell. The lower temperatures also has a negative impact on enzyme activity, so a decent decrease in enzyme influences the reaction rates. So for example, this is one thing that why working with psychophiles is so difficult, is because they are such slow growing organisms. So for example, lowering the temperature by 10 degrees can cause a 2 to 4-fold decrease in enzyme activity, which again can affect growth. So they're really hard to grow with. In their timescales that you're looking at to do experiments with psychophiles can be months to years. Now, the psychophilic enzymes are often characterized by high flexibility, which allows them to interact with their substrates and it lowers their energy needed for these reactions to happen. Another problem, if we look outside of the cell, so within the cell, we've got this, so they have the problem with freezing, but outside, the temperature becomes frozen, as I mentioned previously. And this causes the membrane to become highly rigid and not very permeable. So what they do, they have a number of these molecules within the membrane which allows solutes to transfer across the membrane. Another key parameter that we need to consider when we think about habitability is pH. So when we think about pH, just remember what it is by definition. It's a negative log scale of hydrogen in solution. So each pH unit that we have represents a 10-fold change in hydrogen ion concentrations measured in solution. So when you take it in that context, changes in pH units can actually be really huge. So if you're in an acid environment, it gives hydrogen to the environment, and if it's in base, it decreases the amounts of hydrogen in the environment. So we think about this from a microbial physiology perspective. This is really important. We talked about the respiratory chain and how ATP is produced, which is the driving force for cellular processes, and it's all about this proton moting force, so a gradient of protons across the membrane. And this is something which, as a microbe living at extremes in pH, is really problematic. And typically what microbes that can survive at extreme pHs do, which is different to ones with temperature, is that they moderate their internal pH. So although they may live in extreme environments, what they do is they maintain their internal pH around circumneutral environments. So let's look at acidophiles. So acidophiles are extremophiles that can live in really acidic environments. So if we look in nature, we have acidic thermal pools. A lot of work has been done more recently looking at microbes in acidic acid drain lineage. They can live in environments below pH 3, but they do maintain their cytoplasm near neutral pHs. So what they do is they maintain their neutral pH, but they still have that gradient across the cell membrane. So there's a number of mechanisms that they employ for this. To start with, they have their cell membrane acts as a primary defense against this. So what happens is it can repel some of the protons because the composition of the phosphates, and not allowing them to come into the cell. There's a number of proton transporters which extrude protons from the cell into the environment, which can help to maintain the cells. They also can use the gradients, organic acids within the cell. If it does become acidic in there, you can use this kind of buffering system, which can allow the protons to be buffered, ensuring that the pH within the cell is not demonstrating to some of the protons the enzyme activities that are in there at the moment activated. And at the opposite end of that, we have the halophiles, and these are microbes that can grow above pH 8. We have facultative alkaphiles, that microbes that can grow from pH 8 upwards, and then we have obligate alkaphiles, which need to grow above pH 9 or 10. And the highest values that we have is identified as between pH 12 to 13. And again, alkaphiles there's a whole array of micro environments where alkaphiles can be found. We have the soda lakes, we have alkaline hydrothermic vents. So let's look at how they can adapt. So there's a number of ways in which that they've adapted for this to happen. One way is that they take up protons within the cell by using these kind of antiporters. So they transfer sodium out of the cell and replacing it with hydrogen. This allows a level of protons to be incorporated in the cell. A lot of the work that has been done on bacterial alkaphiles has been done in Bacillus pseudothermis, which is what we have on the diagram here. And what we think what happens, though, the proton mounting force in these environments for alkaphiles isn't conducive for ATP production because of the amount of protons on the outside is very limited. But what we find that they do is they form this kind of microenvironment at the interface between the membrane and the cell, which allows ATP production to still be occurring and driven by protons. So it still produces this proton mounting force across this membrane by regulating its local internal environment. But it also has, again, like acidophiles, it has these kind of buffering compounds within the cell, which allows a kind of buffering to ensure that the pH within the cytoplasm is maintained circumneutral as well. Okay, so let's move on a little bit to helophiles. Now, helophiles are salt-loving extremophiles that live at high salts. Here's a picture of some salt flights in India that one of my postdocs took recently. You know, they can be found in many environments, from hypersaline lakes, such as the Great Lakes, to the Dead Sea. And what we know, the problems that the cells have in these salt environments is due to osmotic pressure. So, for example, if you have a cell and you put it in a really saline environment, such as having an example in the document here at the bottom figure here, what you find is that the water and within the cell, due to hypertonic solution, is released into the environment, which will cause drying out of the cells, which is a problem. And this is something, as helophiles, you know, microbes that live in these really salty environments, there needs to be consideration, because this is a prime problem. If you have a microbe and you put it into a salty environment, the salty environment would call this kind of hyperblock, whereas helophiles are adapted, so this isn't such a problem. We have helophiles that can live up to 6 molar, and we have extreme helophiles that can live above 2.5 sodium chloride. And they've adapted to ensure that water is not lost due to as a result of osmosis. So how have they done them? Well, the helophiles, there's two mechanisms. There are 2 strategies which the helophiles have identified, which they use to ensure that they have this cytoplasmic, and they can maintain proper osmotic pressure within the cell. So if we look here on this diagram, if we take a non-halophile, so kind of what I've talked about before, you put it into a salt solution, all the water is released out of the cell, ending up causing the cell to collapse. So the helophiles have adapted to do this. So let's start. So if we look on the middle diagram, where we have this extreme helophiles, and this is kind of called the salting in strategy, and this is used predominantly by obligate archaeal helophiles. And what they do is they take in some of these salts, and they've adapted to survive in these conditions as well. They also have this kind of strategy for adaptation, where this is more where moderate halophile organisms, what they do is here that they produce molecules such as glycerol and ectidine, which help to work as osmosis solulants. So they keep the balance across the membranes regulated, and this ensures that it excludes as much salt from the cells as possible. So in contrast to what the extreme helophiles do, what it does is it actually doesn't let salt into the cytoplasm at all, as much as possible. Another group of organisms which are particularly relevant when we're thinking about parameters, and I think radio resistance isn't traditionally thought about as much, but it is when we think about it in the context of places like Mars, is radio resistance. And these are microbes that can live in environments that are exposed to high levels of radiation. So they've been found in a number of extreme environments. So Atacama Desert, a number of deserts, including Atacama Desert, are prime examples where we have found and isolated radio-resistant organisms. Normally these microbes are found in kind of endoliths. So if you look at this diagram here on the left, endoliths are kind of microbes that are embedded within the rock matrix, and this allows for a degree of protection from the environment itself. You'll note that in these endoliths are all green organisms, so phototrophic organisms, so they use energy of the sun for life, because it's a highly exposed environment. Examples of these microbes that have been isolated is Crocus idioptis, and as you look at the structure here, what you'll see is that they kind of form these kind of globular shapes. So they're kind of without looking at the physiology internally of the cell, you can see that they're kind of using these mechanisms to help survive. So firstly, they're living in rock environments, which protects them. Within the cells themselves, they're actually protecting themselves with shielding through the structure in which they can grow. Now, the major problem microbes have when they grow in high UV environments is damage to DNA. Mutations caused by radiation is a major problem. So microbes have produced a number of strategies to mitigate the effect of DNA damage. This includes producing photoprotectants, kind of like sun cream molecules, which kind of like the first line of defense, such as carotenoids, some of you may have heard of, and they kind of act like, yeah, as I said, a screen event. And then the second set is, if that all fails, is to have DNA repair proteins. The most damage caused to DNA is through the reactive oxidation species. So what happens is the damage caused by that. So what they have is they have a number of mechanisms which allow them to repair the DNA if damage does occur. Okay, so there are lots of different extremophile environment out there in the environment, and it is really important to think about these when we think about astrobiology. But I think particularly in the relevant for astrobiology are microbes called polyextremophiles. Now, these are microbes that can live in more than one extreme, and studying these can really help to delineate the envelope of habitability and kind of help to develop our theories about where life could live on Earth and beyond. So what are, so let's look at some prime examples. So we've talked a lot about pH and temperature. So here's an example of diagram here of some of these, where these microbes can live. So for example, if you have, there's a lot of work done on microbes that live in high pH and temperature. These are thermophiles, and opposite end of the scale, you have psychoacidophiles, which is low temperature and low acidic. On the right here, I have, there's a diagram which has been taken out of the paper, which is mapped on some of these parameters of pH and temperatures. And what is interesting is this, that you can see that there seems to be the impact of pH and temperature has an impact on where these microbes can be found. So let's look at the impact this has. So if you think about pH and temperature, there's going to be a bit of contrasting requirements with the physiology. So think about the membrane. As temperature increases, fluidicity increases, and in psychophiles, and you'll have a less permeability to protons. And this impermeability is advantageous to microbes that live in high pH and high and low pH, because it maintains the cytoplasmic pH at kind of a neutral pH. So although, we'd expect acidophilic psychophiles and alkophiles to be found, not many of those have been found to date. And this could be due to the fact that there's not as many of these environments, or it could be due to the fact that physiologically this isn't happening. But in theory, this is something that would be beneficial. And if we look at the other spectrum at high temperature, where the membrane fluidicity would increase the protein permeability, which is going to be problematic to microbes that are growing at high pH and high alkaline environments, we do find examples of archaea, particularly at some of these high, low pHs. But what we do find is a lot of these there's not many acidophilic thermophiles that can grow above 100 degrees. And most of these acidophilic thermophiles are actually archaea. So by combining these kind of dual environments, we could include other parameters into this, such as salinity. We can start to really define what life can exist on Earth. So here's a diagram taken from a recent paper, which looks at this in the context of astrobiology. So the diagram at the top left is where we can define the parameter space for life on Earth. So the kind of turquoisey colour shows where culture-based, so we've actually grown these microbes, and the kind of light green is where we have shown microbes can exist using molecular techniques. And then we can look at that in the context of other environments. So obviously this is just looking at pH and temperature, and it's not taking into conditions such as radiation or the availability of nutrients, etc. But if we can map what we know about life on Earth in different environments, we can kind of start to see that actually maybe the feasibility of life exists, that the habitable space in which life can exist on Earth is comparable to that on other planets. So if we take the icy moons, for example, so let's look at Enceladus and Europa, some of those conditions, that there could be a sweet spot in which life could actually be habitable, where habitable conditions that live within could survive in these environments. So let's look at Mars in more detail. So when we think about Mars, we think about the present day conditions on the surface of Mars. They're very cold, they're very dry, and exposed to high levels of radiations. And these these are inconducive to life. But we've talked about endolithic microbes protected from the surface. There could be microbes in the subsurface environment of Mars which could survive and live. And when we want to think about also from a habitability perspective, early Mars, when we look where the conditions could have been more conducive to a warmer climate and denser atmosphere, we start to think that potentially microbial life could have existed in these environments. An example, we look at, the work that's been done on Gale Crater, which has suggested that there were fluvial systems within these environments, which potentially had the building blocks for life, has the key elements that could for life, and also the conditions could have been conducive for this. So if we want to start thinking about habitability elsewhere, we can start to think about looking at Earth. And for that, we can use stuff called analog site, environments called analog sites, where we can start to look where the parameter space is a relationship, and then start to think about this in the perspective, in the idea of habitability beyond the Earth. So a prime example of this is, for example, some work you could look at, for example, the Canadian High Arctic. The environment here, it's really highly saline, it's cold, it has an average temperature about -50 degrees. We know that life can exist within these environments. And what we can do is kind of use these for models to look at potential habitability elsewhere. So for example, work in this site, we could look at using that as a model for looking at some processes on early Mars. So for example, as the environment on Earth, on Mars evolved from these log fluvial systems to the dry conditions that we had today, towards the Noachian Hesperian transition period, there would have been conditions where the environment was acidic, it's not acidic, saline, highly sulfate, kind of cold environments. And we can start looking at analog sites to kind of infer what if potentially life could have existed in this environment, what life that could be. And now this is really important if we want to look at interpreting data from missions and what biosignatures, so what evidence of life these environments, microbes could live. So an example of some work about this is looking at, for example, Gale Crater, Colour Peak, which is a really saline rich environment. And there's been a lot of work done on this over the last couple of years, and it's been used for an analog site for Lake Oakhon period. And studying on these sites, we can start to have an understanding of what kind of microbes can live in these extreme environments. And from this, we can start to infer what kind of community could grow and how these would evolve potentially over time. So interestingly, these are very sulfate-rich environments, and what we find looking at molecular data from these sites is that microbes within these environments would be using sulfur oxidation as a form of metabolism. It's very sulfur rich. We know that in the subsurface environment, it will be very dominated with this, and we can infer from looking at the molecular makeup of these sites that it would be very sulfur rich in composition. And what we can do is using this information, we can then infer that to looking at conditions and environments on Mars. So for example, we can use the data from MSL and use thermochemical modeling to predict what the brine chemistries could have been like in some of these environments. And from that, what we can do is then we can infer using the redox, using Gibbs free energy calculations from the redox oxidation reactions, we can infer what kind of metabolisms could exist in these environments. You may ask, why is this important? Well, it gives us an understanding of what to look for if, with mission data. So, for example, looking for potential evidence of life where there's environments which are rich in sulfate could be conducive to help infer some of this mission data as well. So it's really important to get kind of an overview of these sites. So we use extremophiles to start to think about some of these processes and understand habitability and the boundaries of habitability. But also it's really important for looking at biosignatures and what biosignatures we can use. So most of the work that I've spoken about is extremophiles on Earth, but there's been an extensive amount of work that has looked at microbial survival, not growth, but survival in low Earth orbit. I think over the last 50 years, there's been over 1,000 microbial species that have been exposed to the conditions in low Earth orbit. These conditions differ to what they are on Earth. The major impact that we have is high levels of cosmic ionization radiation, which is something that we can't simulate on Earth. So it's really, the ISS experiments are really important for studying this. A number of missions, international missions, have been looking at the impact of these conditions on extremophiles. So we have the BioMex, and we'll have the BioSign mission soon. And these are basically looking at the impact of these conditions of simulated conditions. So what we do is we're able to use filters. So we look at the whole exposure to the space conditions, and then we can use filters to simulate conditions on the surface of Mars. And this allows us to look at how these environmental conditions can impact the structure and integrity and the biosignatures, or the molecules of these cells. So a prime example of this is some work that is ongoing at the Biomax project. This is some work that Jean de Veer at DLR has been leading, and using a combination of extremophiles with environmental simulations on Earth and in low Earth orbit is kind of bringing up together a database of biosignatures which can be used for looking and detecting life. So for this, it's a whole selection of microbes, extremophiles. So we have photosynthetic organisms. So for example, Crocus idioptis, which has been the one I mentioned about from Antarctica, has been included in this, and some of our hemolithotrophs are involved and have been exposed to these conditions to look at the impact of these conditions have on biosignatures. And I think that this is really important as part of an ongoing project, which is looking at producing a database where we look at these molecules for inferring data. So this has particularly been looking at the Raman and looking at how these signatures are impacted by exposure to these conditions. So this is an ongoing experimental work that's ongoing at the moment, but I think it just highlights how using extremophiles is really important in astrobiology, not only looking at defining habitability, but also for inferring for life, understanding what biosignatures can be used for future life detection missions and what we can expect from the instrumentations and what these biosignatures would actually look like. Okay, so thank you for listening to me for over an hour talk about one of my favorite subjects, extremophiles. But I kind of think that the take-home message here is that extremophiles are really diverse and they can adapt to some of the most extreme environments on Earth. And I suppose the key take-home message for astrobiology research is that actually understanding these and understanding the limits of where they can live and how they adapt and what biosignatures or biomarkers that they produce are really important when we're looking at understanding the limitations and potential of life elsewhere in the solar system. Thank you.